Hello everyone. Welcome to the lecture of Plant Molecular Biology on plant tissue culture. In this lecture, I will talk about different types of culture methods with you. I'm Wilson Yong from the Biotechnology Research Institute, University Malaysia Sabah. The term plant tissue culture is commonly used to describe the in vitro and aseptic cultivation of any plant part on a nutrient medium with an artificial environment. The in vitro works are carried out in a test tube, dish, or flask, which are outside the living organism. Hence, plant tissue culture techniques are also referred to as in vitro techniques. Different types of plant tissue culture include single cell or cell suspension culture, embryo culture, callus culture, bud culture, organ culture, protoplast culture, meristem culture, and seed culture. Plant tissue culture is a valuable tool for studying morphogenesis, cell signaling, physiology, and molecular biology, as well as biotechnology-assisted crop improvement. The impact of plant tissue culture technology on agricultural biotechnology is enormous. Two concepts, totipotency and plasticity, are the central concepts to understand the regeneration potential of plant cell culture. Totipotency refers to the ability of a single cell to develop into a complete organism by regeneration, or to give rise to a whole plant, including metabolism, in a suitable given condition. Totipotent cells are cells that have retained the full genetic potential characteristic of the mature organism from which the cells were obtained, and thus enable them to divide and differentiate into a mature plant if placed in the appropriate environment or under favorable conditions. Plasticity refers to the ability of plants to change their metabolism, growth, and development to adapt to and cope with changes in their environment. When plant cells and tissues are cultured in vitro, they generally exhibit a very high degree of plasticity, which allows one type of tissue or organ to be initiated from another type. Following this principle, the whole plant can be regenerated. This regeneration of whole organisms depends upon the concept that all plant cells can, given the correct stimuli, express the total genetic potential of the parent plant. Plant tissue cultures are generally initiated from sterile pieces of a whole plant. These pieces are termed explants and may consist of pieces of organs such as leaves or roots, or may be specific cell type such as pollen or endosperm. Many features of the explant are known to affect the efficiency of culture initiation. Generally, younger, more rapidly growing tissue or tissue at an early stage of development, or called meristematic tissue, is most effective. When cultured in vitro, all the needs, both chemical and physical, of the plant cell and tissue have to be met by the culture vessel, the growth medium, and the external environment. The culture medium is one of the most important requirements for plant tissue culture and is one of the fundamental techniques that make plant tissue culture possible. The success of the culture medium is largely dependent on its chemical composition, and it has to supply all the essential mineral ions required for plant growth and development. The composition of each tissue culture medium may vary for different plants according to their nutritional requirements. Supplementation of plant growth regulators to culture medium is invariably necessary to trigger various types of growth and differentiation. Although the explants may have certain levels of endogenous growth hormones or naturally synthesized growth regulators, it is often necessary to supplement them exogenously to evoke certain responses. The growth regulators are generally required in very minute quantities, and the nature and quantity of growth regulators in the medium is vary according to the variety of plant, nature of the tissue, and also the stage of culture, such as initiation of callus, induction of somatic embryogenesis, shoot differentiation or multiplication, rooting of shoots, and etc. Physical factors such as light, temperature, and pH also have to be maintained within acceptable limits. Some culture types require aeration and agitation to provide sufficient oxygen to plant cells and to increase the availability of nutrients to all parts of the explants. Plant tissue culture is an essential component of plant biotechnology. Apart from mass multiplication of elites, it also provides the means to multiply and regenerate novel plants from genetically engineered cells. Plant tissue culture is associated with a wide range of applications, such as rapid propagation of plants those are difficult to propagate, biosynthesis and production of pharmaceutical, medicinal, and other industrially important compounds, production of disease-free and resistant plants, germplasm conservation, and so on. When cultured in an appropriate medium, usually with both auxin and cytokinin, explants produce an unorganized, growing and dividing mass of cells called callus. It is normally composed of unspecialized parenchyma cells. During callus formation, there is some degree of de-differentiation, both in morphology and metabolism. De-differentiation is the phenomenon of mature cells reverting to meristematic state to produce callus. It is a transient process by which cells become less specialized and return to an earlier cell state within the same lineage. This suggests an increase in a cell potency, meaning that after de-differentiation, cells may possess an ability to re-differentiate into more cell types than it did before. One major consequence of this de-differentiation is that most plant cultures lost the ability to photosynthesize. Redifferentiation is the ability of the callus cells to differentiate into a plant organ or a whole plant. Manipulation of auxin to cytokinin ratio in the medium can lead to the development of shoots, roots, or somatic embryos from which whole plants can subsequently be produced. Callus cultures can also be used to initiate cell suspensions, which are used in a variety of ways in plant transformation studies. Callus cultures broadly fall into two categories, compact or friable. They are produced when explants are cultured on the appropriate solid medium with the correct concentrations of auxin and cytokinins. Auxin 2,4-D is commonly used to induce callus formation because it can revert cells in the explant to a dedifferentiated state and begin to divide. The cells in compact callus are densely aggregated, and the callus can contain several small prominent embryo-like structures, while the cells in friable callus are only loosely associated with each other, and the callus becomes soft and easily breaks apart. Friable callus provides the inoculum to form cell suspension cultures. Callus proliferation can be maintained more or less indefinitely in the culture if the callus is periodically subcultured in a fresh medium, but they can lose their ability to re-differentiate after prolonged in vitro culture. Callus formation from an explant occurs in 3 stages. During the induction stage, metabolism is stimulated and the cells in explant begin to de-differentiate and divide. Cell size remains unchanged. In the proliferation stage, cells divide actively and the cell size decreases. Cell division is mainly periclinal and occurs towards the periphery, giving rise to wound cambia cells. During the final stage, cells differentiate by expansion and maturation. Vascular nodules or meristemoids are formed in the callus from small groups of meristematic cells. These meristemoids resemble vascular bundles and consist of xylem, phloem, and cambium. The nodules may form root or shoot primordia or embryos. When friable callus is placed into a liquid medium, usually the same composition as the solid medium used for the callus culture, and then agitated, single cells or small clumps of cells are released into the medium. Under the correct conditions, these released cells continue to grow and divide, eventually producing a cell suspension culture. Agitation of the medium serves at least 3 functions. First, it exerts a mild pressure on cell aggregates, breaking them into smaller clumps and single cells. Secondly, agitation maintains uniform distribution of cells and cell clumps in the medium. And lastly, movement of the medium also provides gaseous exchange between the culture medium and culture A. Cell suspensions can be maintained relatively simple as batch cultures in conical flasks. They are continually cultured by repeated subculturing into fresh medium. This results in dilution of the suspension and the initiation of another batch growth cycle. After subculture, the cells divide and the biomass of the culture increases in a characteristic fashion until nutrients in the medium are exhausted or toxic byproducts build up to inhibitory levels. This is called the stationary phase. If cells are left in the stationary phase for too long, they will die and the culture will be lost. Therefore, cells should be transferred as they enter the stationary phase. It is also important that the batch growth cycle parameters, such as the number of cells, the concentrations of nutrients or metabolites, and the cell viability, are determined for each cell suspension culture to ensure the reproducibility of production. Potential uses of callus and suspension cultures include synthesis of important biochemical or plant metabolites by forming a large cell biomass, rapid clonal multiplication and formation of plant species, genetic engineering and production of transgenic plants, somatic hybridization, and development of rapid mutations for resistance to various environmental stresses, including pests and diseases. Organ culture is a type of tissue culture that involves isolating an organ for in vitro growth. Different types can be established, such as meristem culture, shoot tip culture, root culture, anther culture, and etc. Often, any plant organ can be used as an explant for the culture process. With organ culture, or as is with their various tissue components, the method is used for preserving their structure or functions, which allows the organ to still resemble and retain the characteristics they would have in vivo. The new growth continues given that the organ retains its physiological features. As such, an organ culture helps provide information on patterns of growth, differentiation, as well as development. Root culture can be established in vitro using explants of the root tip of either primary or lateral roots, which can be cultured independently on relatively simple media. Since roots are indeterminate organs, their growth in vitro has the potential to be unlimited. Root culture can rapidly produce a large number of genetically uniform roots, making it ideal for studies on lateral root and bud formation, and initiation of cambial activity and nodulation. Although the establishment of root cultures was one of the first achievements of modern plant tissue culture, they are mainly used for the production of useful substances and have less impact on other areas of studies. For example, hairy root culture is a type of plant tissue culture that is used to study plant metabolic processes or to produce valuable secondary metabolites or recombinant proteins, often with plant genetic engineering. The tips of shoots, which contain the shoot apical meristem, can be cultured in vitro, producing clumps of shoots from either axillary or adventitious buds. This technique is also known as meristem culture and is extensively applied for clonal propagation in horticulture, agriculture, and forestry. Because of the small size of the propagules in culture, the in vitro propagation technique is also known as micropropagation. The process of micropropagation can be divided into three different developmental stages. Stage 1. Establishment of explant aseptically. Stage 2. Multiplication of propagules by repeated subcultures on a specific nutrient medium. And Stage 3. Rooting and hardening of plantlets and planting into soil. Embryo culture is a type of plant tissue culture that is used to grow embryos isolated aseptically from the birth of maternal tissues of ovule, seed, or capsule under aseptic and controlled physical conditions in glass vials containing nutrient solid or liquid medium. In embryo culture, the plant develops directly from the embryo or indirectly through the formation of calyx and then subsequent formation of shoots and roots. This technique has been developed to break seed dormancy, test the vitality of seeds, production of rare species, and haploid plants. It is an effective technique that is employed to shorten the breeding cycle of plants by growing excised embryos and results in the reduction of long dormancy period of seeds. Plant regeneration is the major outcome of plant tissue culture, which is based on the principle of totipotency. The ability to regenerate entire plants from selected or genetically altered cells or tissues is the key to the broad biotechnological potential of plant cell cultures. And in vitro plant regeneration is a process in which explants, after undergoing cell division and differentiation, form organs and tissues throughout their growth period. In general, there are two different methods of plant regeneration in vitro for propagation in practice, namely organogenesis and somatic or non-zygotic embryogenesis. Organogenesis is the formation of organs such as leaves, shoots, and roots on a plant organ, usually of a different kind, which is based on the inherent plasticity of plant tissues and regulated by altering the components of the medium. The methods of plant regeneration via organogenesis depend on adventitious organs arising either directly from explant tissues or indirectly from an intermediate tissue, the callus. Both direct and indirect regeneration pathways give rise to meristomoid, and the meristomoid respond to factors in the system to initiate a primordium, which induces new tissues such as shoot or embryo, depending on factors such as different ratios of hormones. In particular, it is the auxin to cytokinin ratio of the medium that determines which developmental pathway the regenerating tissue will take. It is usual to induce shoot formation by increasing the cytokinins to auxin ratio of the culture medium, and vice versa for root initiation. An intermediate ratio of auxin to cytokinin causes the proliferation of callus. Somatic embryogenesis can be defined as the process of embryo initiation and development from vegetative or non-gametic cells which differentiate as bipolar structures mimicking zygotic embryos. Somatic embryos are formed from plant cells that are not usually involved in the development of embryos, such as ordinary plant tissues, and they are not surrounded by endosperm or seed cord. The bipolar embryos germinate to form both root and shoot in the manner of zygotic embryos, usually very rapid, enabling the production of large numbers of propagules. The induction step involved the reactivation of cells to differentiate and develop embryos, and it is achieved by two methods: direct embryogenesis and indirect embryogenesis. Direct embryogenesis involves the development of embryos directly from the cells of explants without any intermediary stages, such as callus formation, whereas indirect embryogenesis involves the formation of the somatic embryos by repeating several cycles of cell divisions and involve an intermediate step of callus formation. Somatic embryogenesis is a multi-step regeneration process that begins with the induction of pro-embryogenic masses and continues with the formation and maintenance of somatic embryo, maturation, and regeneration. Several signaling molecules known to influence or control the formation of somatic embryos include extracellular proteins, arabinogalactan proteins, and lipocyto-oligosaccharides. Temperature and lighting can also affect the maturation of the somatic embryo. In conclusion, There are a number of tissue culture methods since different types of tissues need different conditions for the culture process to yield desired results. Plant tissue culture may be used for cloning, genetic modification of a specific plant, or simply to accelerate or increase the yield of the plant of interest. Although tissue culture is a complex process that requires a great deal of caution to avoid contamination, but it is an investment that could offer rewards once you see the results. With that, I would like to conclude my presentation. I hope you find the topic interesting. Thank you very much for attending and participating in this lecture. Thank you.